Our overall goal was to identify responding CAR T cells, or in other words, those that likely mediate therapeutic efficacy after CAR T cell infusion for large B-cell lymphoma. To do this, we set up to do clonal tracing using endogenous T-cell receptor sequence as a barcode to trace CAR T cells through time. With this work, we defined infusion product CAR T cells that expand clonally during CAR T cell therapy in patients for lymphoma. And in the work, I present specific gene expression programs and transcription factors that define this cell population. In addition, during our work, we observed that several cell populations expand during CAR manufacturing, as well using clonal tracing. And we found that it is a mix of naive central memory and helper T cells that expand during manufacturing. In addition, to our surprise, we also observed that T regulatory cells expand clonally during manufacturing. And that of course, brought our recent work on linking CAR T regulatory cells to poor clinical outcome in the context of large B-cell lymphoma. And with multiple methods, we were able to link the post-infusion CAR T regulatory cells that are linked to poor patient outcomes to these pre-existing T regulatory cells that are also linked to poor patient outcomes and are also elevated in patients with large B-cell lymphoma. Together, our work provides insights into CAR T cells responding during CAR T cell therapies, as well as nominating removal of these pre-existing T regulatory cells as a potential approach to improve efficacy of CAR T cell therapies for large B-cell lymphoma.